Hello, growers from around the world. Jordan River here with more Growcast. Thank you so much for tuning in today. Another new guest. Today we have Taylor from Rimrock Analytical on the line. Really interesting episode. You guys are going to love this one. I'm not even going to set it up. I just know that you guys are going to absolutely love today's episode. Before we jump in with Taylor, going to give some love to our proud partners, Plant Success. PlantSuccess.com. Put a hyphen in between those words. Go ahead and check out their Great White Myco. Go and try their King Crab. I'm hooked. I'm hooked on the crab. I think their Myco Chum probably gets the least amount of play, which I recently learned has a little bit of molasses in it, I believe. And I'm using that Myco Chum because I like to stack my microbials, and they all gobble up that Myco Chum, baby. And don't sleep on their Orca, Liquid Mycorrhiza. The Plant Success line has what you need. Get it in your local hydro store. Check it out online, Plant Success. And be sure to check out the episode that we did with Jim Egan, of course, so you can learn more. Proud partners, so thank you to Plant Success. That's all for the intro, everybody. Let's jump in with Taylor from Rimrock Analytical. Thank you for listening, and enjoy the program. Hello, podcast listeners. You are now listening to Growcast. I'm your host, Jordan River, and I want to thank you for tuning in today. Before we get started, please share the show. Spread Growcast around. Tell a grower it's how we grow. Make sure you're subscribed. Give us a rating and review, would you? And hit up patreon.com/Growcast for all the mounds of bonus content. Today, we have a very fascinating episode. A new guest on the line from Rimrock Analytical. We have Taylor. What's up, Taylor? How you doing today?
Hey, Jordan. I'm doing well. Thanks for having me on the show. Pleasure.
Thank you for coming on the show, man. We're happy to have you. I love learning about cannabis science, cannabis testing, all of that stuff. And Rimrock Analytical does some really unique and interesting work. Before we dive into all of that, can you give a little bit on your personal background? What brought you into the world of cannabis and eventually cannabis testing and analytics?
Yeah, absolutely. So kind of a meandering course through my career. I started, academically, up in molecular biology, chemistry. I was doing a lot of genetic analysis of things. And I started my career in cancer research. Did that for a number of years before kind of moving out of there, with grant funding and kind of the monetary side of things. It was kind of a challenge to make a real go at it. So I ended up in industrial water treatment. We're doing recycling, reuse applications for large industrial processes, and I've really done that. That trend has kind of carried through the bulk of my career. About 5 years ago, I got in with a company, Silver Bullet Water Treatment. And that's really where my introduction to the cannabis industry, the cannabis community really kind of kicked off. And,
Nice.
About 2 years ago is when we founded Rimrock Analytical. And that was really just out of the marriage of, my passion for analytics and particularly kind of the genomic and microbiology side of things. But also just realizing this need for greater accessibility for the community for this technical data that's out there, but a lot of the larger organizations and things, it's not that accessible to a lot of people. So we really wanted to open up those doors and that data to people.
Man, the more data, the better. I think that's a huge forefront in the cannabis industry. I look at these companies like yours that are just trying to help people understand their grow better so they can optimize things and keep things in line. But man, I have so many questions about, do you just want to jump right into testing and like what the testing processes look like and what we all test for? Does that sound good?
Yeah, absolutely.
I would love to start with you. We were speaking off air and you were saying that one of the major players in your business is testing for like pathogen identification, like microbial identification. Can you talk about that?
Yeah, absolutely. So, we've come a long ways in terms of genetic analysis. If you think back, like the Human Genome Project was this huge groundbreaking effort, multinational, major resources invested in this. And,
Right.
That really wasn't that far ago. I had an interesting conversation with some of the molecular diagnostic instrument vendors. And, they're talking about it. It's like, what would it take to do the Human Genome Project today? And they're like, yeah, a single lab could do it in less than a week.
Wow.
Or, less than, a few $1,000s. And it just blew my mind. And so it's basically taking the advents that we've had in this molecular science and, how can we use that in a way, that's beneficial to the cannabis community? And one of those ways we've found is identifying pathogenic microbes, particularly early on, before you see onset of pathology or onset of disease. That's critical. It's useful in, validating your IPM strategies, your biosecurity, just an ounce of prevention, right?
Yeah, absolutely.
So, we're doing a lot of that. We're also doing, use the same technology to quantify beneficial microbes. That's huge now. People are looking at different ways to apply, beneficial mycorrhizal organisms. But how do you know, they're doing their job? How do you know how persistent they are in the root zone? How often do you need to reapply these products and things? So,
wow.
You can look at it on both sides of that coin.
That's incredible. What does the identification process actually look like? Like, is someone looking through a microscope and it's like identifying, like a arachnid versus this one has 6 legs, or, this amoeba has like 8 stems, or is it more chemically tested, digitally tested? What does that look like?
Yeah, so there's a number of different platforms to use. PCR is one of the dominant ones, in different kind of flavors of PCR. So PCR stands for polymerase chain reaction. It's a molecular process where you can take really small amounts of starting target genetic material that's very specific. So say you're looking for Fusarium oxysporum, even more specifically down to the species level. You can put out a targeted piece of genome that's unique to that organism. You can amplify it to a huge amount. And then you can use detectors downstream to tell you that it's there, essentially, and you can actually quantify that. So that's where you get your sensitivity, but also your specificity. It's very, very specific, so you get really accurate results in that way.
Whoa! So it is a digital readout from as you said, a molecular reading instrument.
That's right, and there's other ways too. There's things. I won't go too far into it, but there's microarray analysis. There are just Sanger sequencing analysis. There's a thing they call LAMP, which is like a isothermal amplification, where I know it's very jargonous, but
no, I love this.
There's a lot of processes out there, and that's one of the things that's really exciting to me is this is relatively new in the field of cannabis, in the sense that we have all these tools in our toolbox, but what are the best ways? What are the most responsible ways to apply this to the cannabis industry to meet our needs?
Wow. Can you answer that question? What are some of the ways you're changing growers' lives? Are you sending back devastating results sometimes, saying, listen, you have like a horrible pathogen mold, you need to do you're wasting your time, do a restart? Are you mostly optimizing people's grows, a little bit of both?
Yeah, it's a mixed bag. I mean, it's been a unique vantage point we've had, kind of peeking behind the curtains, so to speak, where, we've seen things where, pathogens are relatively ubiquitous, actually surprisingly so, I've found. We'll see, certainly not at every grow, but we'll see things like Phytophthora or Fusarium or Pythium, they'll pop up relatively frequently.
Wow.
The interesting thing around that, though, is much less frequently do you see the disease. These plants are pretty robust.
Wow.
And they've kind of co-evolved through the ages with these organisms. So that, coupled with, we talked a little bit about the beneficial microbiology that kind of combats these, that's antagonistic to these pathogens. It's less frequent that we see that really sad plant that's, all yellow and wilted, and, their roots have turned into brown snot. But, that does happen from time to time. It's unfortunate. A lot of times that's a restart situation.
There's a pathogen completely gotten out of control. That's wild, man. Super, super interesting. But like you said, they're kind of everywhere. Just like viruses in humans, we evolved alongside all these viruses, and we're carrying all sorts of different, we're basically a host. And you're saying the plant is too. It just usually doesn't show symptoms unless something has gone wrong.
Yeah, and a lot of times the pathogen just doesn't have that chance or that opportunity, at least in an ideal scenario,
right, to take over.
Exactly. Your biosecurity plan is really your frontline defense. Let's keep these things out to the best of our ability, but understanding these things are everywhere. Especially a lot of these grows. You have people coming in and out. You got some transfer potential there. Once they get in the facility, it's really difficult to completely eradicate them. So your next step then is, how can we get these plants very resilient to show the disease, and they're pretty good at fighting them off for the most part.
Wow.
You keep them happy, you prevent that stress. You get your beneficials in there, and really you've got a good chance to prevent that disease.
That's wild, though. It's a different way of thinking about your grow. Can the transmission be all sorts of things, from a spore being on you to insects biting one plant and then biting another?
Yeah, absolutely. They're vectors, which are living things. You mentioned insects and fungus gnats and things like that, bugs. And there's another term, fomites, which is more inanimate objects. So think of like tools and shoes and clothing. People moving in and out.
Right.
These are two different primary transmission modes.
Wow, that's wild, man. One thing that's been coming up on this show, not too recently, but has in the past quite a bit. There are some strange viroids out there that people occasionally post online, and some we understand less than others. There's the hops latent viroid that gets into cannabis. There's also this dudding syndrome that I don't know if that's the same or a different strain or a completely different viroid, but this idea that these plants are getting some sort of viroid and their potency just goes to shit. It's like dud, bud. It's dudding. Can you speak on any of those viroids, and are those things you test for?
Yeah, they are. And this is a great example of where those molecular diagnostic tools come into play. Because bacteria, fungus, things like that, there is the option to do a selective culture media, which is your petri dish, and you can quantify and identify them that way.
Right.
Whereas these viroids, they're purely genetic in nature. They're just this like rogue stretch of RNA that gets into these plants, and as you said, they've been commonly called dudding, where your plant just it doesn't look sick, but it's just small. Its yield is way down. It maybe grows in weird ways, has weird offshoots and things. But it's this genetic material that really you need those molecular tools to identify. And viroids are interesting. They're really specific to plants. So they're not to be confused with a virus. So virus is something that, we're familiar with, of course, in this day and age.
Too familiar.
Yeah, it's a little different. Viruses can be DNA or RNA, and they have a protein coating, with different structures on that protein coating. They're kind of encapsulated. Where these viroids they're just naked, like I said, rogue genetic material. Jesus,
and they just bounce off of humans, animals, other plants too. Are they kind of like cannabis specific? Like a lot of powdery mildews are plant specific.
Yeah, there's some specificity. I mean, as the name suggests,
the hops one,
right? It's involved in some other plants and other species. There's a lettuce kind of varietal that also has been known to affect cannabis. But yeah, I mean, you see a lot of transmission through trimming tools, different things where you can actually kind of get into the vascular tissue of the plant. So yeah, it's becoming an issue for sure.
Jeez. That's really wild stuff, man. What about the tobacco mosaic virus? Is that one that you see a lot? Is that something you could speak on at all?
Yeah, I mean, I haven't seen it myself too much. That one will typically show some level of pathology on the plant where you can kind of visually get some cues going, a little bit more so, I would say, than the viroids.
Right.
But again, that's another great one. It's challenging to culture viruses through, what I would consider kind of more traditional methods. I really lean towards those molecular diagnostic tools for that.
Wow. Like I said, it's kind of a different way of thinking. You don't want to be paranoid about this sort of thing, but you do want to clean your scissors and make sure you're keeping pests out of your garden. With these viroids and these I'm not even going to try to classify. With these viroids, is there any getting rid of them? Is it now part of your plant, something you manage? Are some of these, like the dudding, it won't go away? I'm thinking you might want to throw out your genetic stock. Like, what does it look like as far as treatment, and what do you recommend people do when they might have some of these strange viroids?
Yeah, I mean, I've heard of success through some, they'll call it cleaning up your genetics through some
Oh, that's right. Oh shit. You recommend they do a tissue culture? That is so cool.
Yeah, I've seen that. I have seen people kind of just put their hands and be like, look, we need to start fresh here. And obviously that works as long as you're starting with clean genetics as well. But yeah, it's tough. It's not something where you can apply like some sort of application of foliar spray or some sort of chemistry, because it is really embedded in that plant.
Wow.
So yeah, you have to take different routes.
I know you're not a tissue culture company, but What is it about tissue culturing that allows it to shed that harmful RNA strand?
I mean, cannabis and plants in general are interesting in that you can do these clones relatively easily,
Right.
it provided that section of the vascular tissue or that section of the plant is void of viruses and things.
Oh, okay. So if you took like a sick part of it, okay, duh.
Yeah, or a healthy part. You see these pathogens, they tend to kind of accumulate in certain parts of the plant. So that leaves other parts potentially cleaner, so to speak.
It's crazy.
So you can kind of clone. I've heard the term "clone out" of a disease or clone out of a problem, an issue with that plant. So the genetics of the plant, all the genotypes, they're conserved. You can maintain that strain, but presumably you can get away from that disease or that pathology.
I never really put together like taking a part of the plant that's not sick. Like it is very simple. It's a lay person's approach, but of course, theoretically you could kind of take that piece and continue with that, not containing the same RNA. Super interesting, man. That's really wild.
Yeah, as long as that disease or as long as that infectious agent is completely systemic, yeah, there is that opportunity.
Wow. What is the majority of your business? Are you usually doing these microbial testing? What comes in second behind the microbial identification?
Yeah, I mean, we do a lot of nutrient analysis. What's really getting a lot of steam lately is plant genotyping for things like early gender identification.
Oh yeah,
particularly in a lot of the breeders and people growing seed.
Oh, it's such a huge time saver.
Yeah, no, it's great. So that's something that we're looking into, and that seemed to be a really popular service. We're really trying to take that deeper sense, though. Our plan here is not just to throw data out the door and here's give us some samples and let's give you some numbers. We really want to be more integrated to the success of these grows, these cultivators. So we're still in partnership with Silver Bullet Water Treatment. I mentioned being a part of them earlier on in the show.
Ooh.
So they're doing kind of the water management side and the design engineering side of things. Rimrock steps in and we can do things like, let's look at what your nutrient mass flow is. You've got your nutrient feed, particularly in indoor grows. This becomes really prevalent where you can track. Let's see what we're adding. What is the plant physically uptaking? You can do tissue analysis, sap analysis, and then what is your runoff? And through this, you can kind of do a mass balance to see how efficiently your processes are working.
Wow.
I've heard it termed pipe to plant analysis. So it's really characterizing your whole system. What we find is to no detriment to anyone, but a lot of people tend to kind of overfeed. They tend to be kind of on that side of more is better mentality in terms of feeding plants, feeding water, which you can get by on that just fine to an extent, of course. But what we're finding is, there's a lot of efficiencies to be gained, not only from, your process and your cost that you're investing in, whether this is your job or it's your hobby, there's a lot of investment there, but also, the world we live in, we're really looking more at sustainable strategies. How can we make this better where you're not sacrificing your end result, you're not sacrificing your yield or the quality of your plant, but you're also doing it in a responsible way where you're managing the amount of inputs, you're managing your water resources, your nutrient resources, your waste. You add more nutrients, more water, conversely, you're going to have more waste to deal with. You're going to have more runoff. And what do you do with that? I mean, you're putting it down the drain. You're doing, some people are recycling it, but that's really the mentality we're looking at this.
Man, that's incredible. Helping growers understand their grow better and maybe garden more responsibly. That is right up our alley. Before we return to Taylor, shout out to AC Infinity, the creators of the greatest grow fans on the planet. ACinfinity.com, code GROWCAST15. Now, we're having some trouble with the Amazon code for AC Infinity. Some people get it to work, some people don't, and we're looking into it, but you can definitely use it on the website. Just go direct, ACinfinity.com. Scoop their tents while they last. Those tents have, I think, the thickest material, or tied for the thickest material you can find on the market, and they are sexy looking, and they even have a little mount for your AC Infinity controller. But if you're using any fans other than AC Infinity, you really need to upgrade. Use GrowCast15 to save 15%. Send us a snapshot of you using our code to be entered to win free seeds, USA only. And we appreciate you guys using AC Infinity, our proud sponsors, and supporting the show. AC Infinity. Upgrade your ventilation game. Get a tent if you can. Like I said, they sell out quick. You won't regret it. Use Growcast15 and send us a snapshot. All right, everybody, let's get back to Taylor. What has all this testing revealed to you about the current state of the grow world? Before we get into some of those other points, what have you learned from doing all this testing as far as like the state of the grow industry and growers as a whole?
Yeah, it's been totally fascinating. I mean, coming kind of from the different varied backgrounds that I have in the analytical world and the testing world, what's really cool that I've found is the industry's hungry for this knowledge. There's really been a high acceptance of, let's get you a deeper dive into your process and your through analytics, right? Mm-hmm. As we're doing this, we're finding these areas of optimization where, people have done things successfully for a lot of years. But that doesn't mean that there's not room for improvement and, there's room for optimization. So that's been really interesting, and it just shows the level of opportunity that's out there.
When it comes to specifically, kind of jumping back to the high feeding thing, organic growers, synthetic growers, do you see these guys running into the same problems? Do you see having to cater to them differently if they're in like a living soil versus like a cocoa with a bottled nutrient sort of thing?
Yeah, I mean, I'm not here to say, one is better than the other. I don't think that's the case. I think, they're both, adequate, or that's not the word. They're both,
appropriate in different situations. That's kind of what we preach on this show.
Yeah, absolutely. And there's really no wrong answers in that sense. But with that being said, I will bring up that there are different approaches that we'll take, not only from an analytical side of things that are important now to monitor based on the type of grows that methods that you're using, mm-hmm. but also on, going back to the sustainability conversation.
Right.
How are you managing that? So, if you're using, what we'll call a synthetic or a salt-based, nutrient formulation, mm-hmm. your reuse is going to look differently than those that are using, these compost teas and these organic,
sure.
feeds, right? And so how do you approach that? If you want to reuse your runoff, for example, that's becoming increasingly popular. Reuse your condensate, reuse all these different water streams.
Oh, damn.
That process is going to look totally different potentially based on the types of not only grow method you're using, but inputs and kind of overall strategy you're using.
Absolutely. That's a great answer. When it comes to trends, do you see, this is something that I'm really fascinated by. I'm going to try to articulate this thought. You can follow up on it and tell me what you think. I know you're not a doctor, but I'm just going to lay this out there because this is something I've been thinking about for a while. I'm involved in the health field. We do a lot of regenerative agriculture talk. I'm really fascinated between the link of so-called health and wellness, and then the link of microbiology, and then the link of sterility. So let me dive in there. There's all of these studies coming out on the importance of your gut microbiome and your skin microbiome and keeping all of this microbiology alive. And it's basically the same exact thing as regenerative agriculture and the rhizosphere. Literally, they play into one another. If our diet is rich in these organic foods, it actually helps our microbiome. And if they're not, and they're covered in pesticides and they're sterile, it harms our microbiome, etc. So there's all this reverse research on, for instance, doctors saying, don't avoid peanuts with your young kids. You need to expose them. It's all about exposure now. Get them in the dirt. It's good for their immune system, all this stuff. Exposure. On the other side of this, I'm in the medical cannabis world. And a lot of these medical cannabis grows are for very compromised individuals, immunocompromised individuals, some of these states where they don't have recreational and they are only catering to sick individuals. They want, quote, clean, healthy cannabis. And that means sterile. That means you're not bringing in manures, which might have God knows what on them, which might be optimally healthy for like someone else. But for a compromised person, there's a big difference to me. There's all these different facets between clean, healthy, and sterile. And it really depends on your situation. I'm wondering from your testing standpoint, do you see more sterile synthetic growers running into the same, for instance, pathogens? Do you see that more often with organic growers? Do you see a difference in like cleanliness between those two grow styles? Or are growers kind of all running into the same problems? I know that was a lot to throw at you, but just give me your thoughts.
Yeah, no, that's great. I mean, that's deep, and I think that's really part of the frontier of not only medicine but just kind of general scientific inquiry. I think it's irresponsible to take the stance of someone from science to say that we know this, how interrelated these systems are very, very
Right.
well. We're constantly learning. I think if you stay on top of the research, there's ever emerging data that suggests that there are significant impacts to disrupting these interrelated systems, the microbiome, and
Yes.
diet, and different sorts of you're talking about, it's a culture of sterility for sure. If your body's not exposed to things, particularly early on, you've got this innate immune system, and it's similar with plants, right? They have this immune system, and if they're never challenged, that immune system just kind of sitting there idle in the background, and it tends to overreact when it does see a stress, and you see these autoimmune diseases, you see all of these different allergies and all these things popping up. So I think it's totally fascinating. Going back to your question on the different types of grow strategies, I would say that they certainly do see different challenges and different issues. So, I was working with a commercial cultivation facility, and they were, I mean, this is like medical grade, like clean room. This place was just immaculate, and it's very impressive to go and see these types of facilities. But there was an upset in their water system.
Oh.
And by that, I mean, just, kind of a relatively subtle change. And their system just completely filled up with biofilm. It just turned into just snot.
Yeah, that's that funk you get on the top of your reservoir. If the biofilm is that nasty shit that forms, it's like algae and stuff, right?
Exactly. And, in this case, it was really pretty devastating. It was plugging emitters, there's a top feed drip type process. And it was really rough. And what we found really is that the system was so sterile that, just the slightest upset to that system, you had that, those microbes in there, and they kind of turned into this defense mode where they generate this, you call it this extracellular matrix. It's this matrix of biofilm in biology, and it just plugged everything up.
Whoa.
With that, coming from a microbiology world, sterility is kind of a misnomer, right? So there are scenarios that places are truly sterile. They're devoid of biological life.
Okay, right.
These are few and far between. I mean, we're talking about pharma and crazy research labs. So when you're talking like a sterile grow room, there's bacteria in your water. There's bacteria on your plants.
Good point.
There's fungus in there. Just because it's not an input to your system doesn't mean that it's not there in some level.
That is a good point.
And so, you can see those upsets. Now on the more organic side, where you're adding these beneficials and things, we run a test with these, and they're teeming with life, right? They're growing off the table with different microorganisms. But at the same time, that doesn't mean that you can't get pathogens to sneak in there, and you can't have these biofilms and things we've seen. That chance is there. I always look at biofilms in the sense that it's kind of a energy calculation for these microbes. Not to personify them too much, but they're sitting there, they're happy, they're doing their thing. You add a stress to that, you change the temperature of their environment, you add a disinfectant, or you mix something up, and you kind of freak them out a little bit, you shock the system. A lot of them, not all of them, but a lot of them have this tendency then to go into protect mode, and they generate this biofilm. You can think, or I think of the biofilm as like, you're erecting a castle, right? You're protecting yourself from this outside stress. And, to no fault to the microbe, that's, they're trying to make a go at things, right?
Trying to survive.
But,
wow.
Trying to do the thing. But that can relate to us, or translate to us as, this is a big nuisance. And once you start to get that biofilm in place, it's tends to attract, and other pathogens and things can adhere to it. They can start to proliferate from there. Obviously, you have the physical hindrance of plugging. I've seen pumps go bad. I've seen emitters obviously plug. So you can really get down the slippery slope.
Could that happen not just in like a water or a feed? Could that happen inside a soil? Is that like when you said a temperature change, and that really peaked up to me. Were you specifically talking about, for instance, when things get too warm? That's generally what OG growers will say to avoid. Kind of a warm, wet root zone is when things turn south. Or are you talking about specifically like water?
Yeah, I mean, my mind always jumps into the hydroponic type of grow, but soil is not immune to these types of things. You even if you're doing a soil grow, you're watering your plants in some way, whether it's hand watering. Obviously that's less of a concern, but if you're using any sort of automation, any sort of feed system, that could be an issue.
True.
Yeah, in terms of temperature is huge. Temperature in the environment is, I would equate that to something it's pretty broad. It's like pH in water.
Right.
It's for as simple as it seems on the surface, it has so many interrelated impacts to the success of your grow and the different processes that are going on. You look and I'm not going to pretend that I'm a cultivation expert by any means, but you look at how temperature fluctuations can affect your BPD and can affect things where you're going to promote mildews and you're going to promote pathology and negative microorganisms to proliferate in your system. So it's certainly a big deal.
Man, I feel like we could go for a long, long time here. I want to make sure we have room for water treatment. This is something that's been coming up on my show like crazy. We do a live web show for members called Growcast TV, and we recently pulled up the EWG, the Environmental Working Group. They have a bunch of really cool tools on their website, but one of them is the tap water database, to where you can see the contaminants in your local tap water, searching by zip code, and the levels that are above the guidelines and which ones are under the levels. Oh my God. It was like all the members now know they have poison water. My water had chloroform and radium. Someone's had uranium. We were making all these Bruce Banner jokes. It was ridiculous, man. But I would love to kind of dig into your mind now that I'm so piqued by this world of water filtration and realizing just how wildly poisonous and different our water is from one another. This country has really fucked our water supply, and it seems like every city and municipality or well or whatever has different toxins. Like, what does your work kind of bring you into the world of water filtration? What does that look like, and how is this transforming the lives of growers when they get their water on point?
Yeah, I mean, water, obviously, I've spent a big chunk of my career as it relates to water and kind of science of water and treatment strategies. It's another one of those things that's taken for granted in a lot of scenarios. You look at water, and one of the questions that's posed is, why do we see such diversity in water quality? Right? It's coming from an aquifer, it's coming from ground, it's coming from a municipal treatment system.
Right?
Why is there such a change? And a short answer to that is, water in a way is considered the universal solvent, and by that I mean it's really good at dissolving things into it. In fact, if you get, people talk about, reverse osmosis, RO permeate, deionized water. It's you're basically removing all the dissolved constituents from it. But what I think is missed with that is on the downstream side. Once you have this ultra pure water, it has a really strong driving force to dissolve things back into it, right?
Wow.
And these things can not only be salts and the physical things that we think about, but it's gases, it's vapors. If you have any sort of volatile organics in that environment, they're really going to soak into that water stream. I did to illustrate this point. It's kind of funny. Back in college, I had shown some buddies of mine. They had their college house or whatever, and as you can kind of imagine, I'm sure you can illustrate this in your mind, the refrigerator was kind of a horror show a little bit. But I gave them this ultra pure water that we pulled from the lab. They had some odors in their refrigerator. I was like, just put that in there and leave it overnight. And sure enough, overnight, the refrigerator smelled better, but you pull that out and the water smelled like pizza and all these things. It had just absorbed all of those, volatile odors into the water. And it kind of blew their mind. They're like, wow, that's crazy. But, kind of getting back to the broader sense of water treatment, you're right. Water quality changes. It's variable. We always from, talking with, the silver bullet side of things, silver bullet water treatment, we start with a water analysis. You have to know what your objectives are and you have to know what your starting material is, right? I think we hear a lot of people just kind of jump to, you just need RO, right? And I'm not here to knock RO. RO is it's an extremely powerful treatment technology, and it's applicable in a lot of places. But I would argue that it's not necessary for all applications, for sure. And so if you know what you're starting with, if you know those constituents, much to what you described here, you looked at, kind of the averages around the country. Let's identify the 2 or 3 or 4 whatever constituents in this water that we see as being problematic. Now, how can we address these straight on? Right?
Exactly.
You don't necessarily need to strip that water down to zero, down to nothing to have a good product. Particularly, and a lot of these growers, you're going to add a nutrient feed of some sort on the back end, and so you're just adding a lot of it right back in.
Yeah, that's true too. And also, you're going to run into some issues where there are some desirable micronutrients in, especially if you're on like a decent well water. So it is all about understanding your source. I spent the first part of my grow career, the real part of my grow career, now I'm just a home grower, in Humboldt County, and the water quality there was so much better than Algonquin that I never ran a filter. I never had any problem. I just dechlorinated and I was good to go. This Algonquin water had, 20 contaminants and 12 above the limits. And the cool thing about this EWG tap water database is if you scroll down, it tells you which filters knock out which harmful components, which is exactly what you're talking about, Taylor. And to be fair, RO, if you look at RO, that's where the most checks are. However, it wasn't everything. There were some things like chlorates in my water that affect thyroid health, and who knows what they do to plants. And that doesn't come out with a reverse osmosis. It shows you what a carbon filter does. It shows you what a deionizer does, all this different stuff. So it really does depend, as Taylor's saying. But my question is, Taylor, I've had a few questions when we did this exploration. The first one being, why are they all so different? And I love how you addressed that. But what about this one? Why isn't there like a universal treatment method? Why haven't we figured this out? Why are some so much better than others? Is it because like we don't have a standardized treatment method? We don't know what works. I mean, I was just stunned that the qualities were also different. We're not treating this is city tap water. They're not going out and testing a river. This is what's coming out of your tap. Why hasn't the nation as a whole kind of gotten a grip on this?
Right. I mean, that's a great question. So I would say that it's certainly not the case that we, and by we, I mean, society doesn't know how to treat the water. I would say that is not the case. There's ample technology and ample knowledge out there.
Jesus.
Very, very deep knowledge into how to purify water, right? And there's certainly been attempts at standardizing what would be considered quality water. We have the Clean Water Act. There's a lot of EPA requirements and AWWA requirements and things like this. Really, what it comes down to is depending on the size of the municipality or the distribution system. A lot of these guys are using RO. Frankly, they're using reverse osmosis on a huge industrial scale.
Whoa!
So you're starting with clean water, but here's the caveat: as I said before, it's the universal solvent. So when you have this ultra pure water, and you intend to ship it miles and miles through piping and all these things, if it's that pure, it's going to pick up things along the way. So often what they do, in ancillary to that, or in addition to that, really doesn't taste that good. If you just have pure, just there's nothing in it, you hear people on taste tests, it's really not that satisfying. So what you'll see these companies do is or these municipalities, they'll backfeed with buffering salts, and they'll backfeed with some things. Of course, there's a requirement for a low residual chlorine. You'll see fluoride and things like that. They're intentionally added back into the water stream to kind of stabilize and buffer that water.
Right.
And then it goes down the distribution system. So the question is, when you're seeing, kind of more scary things in the water, a lot of that can come from the distribution system. A lot of that can come from dead legs. And, there are efforts that are taken to kind of track those and rectify those, but they happen. When you see things like chloroforms and things like that are really scary, these fall into a category that we call disinfection byproducts, particularly with chlorine or hypochlorite chemistry, hypochlorous acid. As they react with different organics in the water and different contaminants, they transform. They change into different critters, and you get things like trihalomethanes and things like chloroform, right? And so those can show up in your tests, and they're spooky when you see them.
My water was making me sleepy, man. I have a glass of water and I need to take a nap. Chloroform in my water. What the fuck?
There you go.
It's unsettling, and it makes you think what it's doing to your health and your family's health, and then your garden as well. We have another cool thing about that website is they show the detrimental effects of these compounds on humans. What is it doing to the plants, I wonder? At the very least, it's some of it's probably making it into the cellular structure and into your flowers. I mean, that's just how it works. I don't know though. I have no idea.
Yeah, I mean, the cannabis is, I don't know if notorious is the right word, but it's a great bioaccumulator.
Right.
And so it you know, in that way you can kind of see a parallel with water. Water wants to soak everything in out of the environment, and cannabis wants to absorb it up into its right?
Christ. This has been a really eye-opening episode, Taylor. I think that there's been a few points you've made that are gonna shift the way people look at their grow. So yeah, I just want to thank you for this eye-opening episode, and make sure that people get at you too. Why don't you throw out your Instagram handle, URL, anything you want my listeners to go and check out?
Yeah, absolutely, Jordan. And again, I can't thank you enough for having me on the show. You can find Rimrock Analyticals, just www.rimrockanalytical.com. Instagram is Rimrock Analytical. Pretty easy to find us. We try to put out educational material best we can. You know, we're out here to be part of the industry, part of the community. So yeah, if you guys have questions, you have anything, reach out to us. Happy to chat.
Go and get it, everybody. Before we sign off, let me ask you what is coming down the pipe? What's in the future? Is there anything that Rimrock is looking to expand into? And if not, any new like technologies you'd like to share? Just anything future facing.
Yeah, I mean, we're always trying to be on the forefront of things, you know, and keep, I had mentioned very early in the show, you know, keep this accessible to people. So as technology advances, typically you see price points and things like that barrier to entry kind of decreases as well.
Right.
And so we're always trying to stay on the forefront, give you the latest and greatest technologies and methods. So yeah, we're certainly keeping our pulse on that and our finger on that pulse. And yeah.
Just further testing, more analytics, more data collection, that sort of thing. Yeah,
definitely. You know, I'm interested more in the genotyping of plants. We've done a lot with the microbiology, but you know, there's new methodologies coming out for like THC, CBD, synthase precursor genes. Looking at environment aside, you know, what is the potential of this plant for producing different cannabinoid profiles? Looking at things like that. So yeah, just taking a deeper look into learning more about this plant.
Listen, I think it's awesome. I love the wide variety of options that you're giving growers. And I urge all my growers, to go check out Rimrock Analytical. We'll throw the name down in the description. Taylor, thank you one more time, man. This is a wonderful episode, and I hope you'll join us again soon. I would love to have you back on.
Absolutely. Anytime. Just you know where to find me.
Perfect. All right, everybody. Thank you for listening, and stay tuned. Got some great content coming your way. This is Taylor from Rimrock Analytical in Jordan River, signing off, saying have an extraordinary day out there, everybody. We'll see you next time. Be safe and grow smarter. That's our show, everybody. Thank you so much for staying subscribed and downloading. I appreciate all of you. Before we wrap it up, have you heard about jackgreensgardenproducts.com? Go and check it out. Use code GCListener for a whopping 25% off your organic soil conditioners. And of course, my favorite IPM product in veg, Jack's Bug Be Gone. This is a pepper ferment. It is my favorite. If you haven't tried it, go and try it from jackgreensgardenproducts.com. Use code GROWCAST25. And the great Joaquin should be making another appearance soon. So stay tuned for that. We really like those guys. That guy. All of them over there. All right, everybody. Hope you enjoyed the program. Quick shout out to Jay Blanked for doing the beat. Quick shout out to membership and all the members. Oh man, just having so much fun at patreon.com/growcast. And finally, quick shout out to Illinois Canna Consulting, the great Rhizo Riches consulting company, illinoiscannaconsulting.com. All right. See you next time, guys. Stay safe out there. Bye-bye.